All right, let's dive in. The ultimate goal, maybe the holy grail, in anatomy is really total visualization.
Seeing the whole picture completely.
Getting that perfect 3D map of,
Yeah.
well, an entire complex organ. Think down to the smallest blood vessel.
And without messing it up, right, without compromising its natural structure. That's the key.
Precisely. Like imagine mapping a whole mouse brain while it's still sitting inside the skull. That hard, bony structure.
And doing it all at submicrometer resolution.
Wow.
That level of detail is just, it's been the dream.
But it sounds almost like science fiction, doesn't it?
Well, for decades, it really has been one of the biggest technical barriers out there. Because when you're dealing with complex living systems, vertebrates, standard 3D histology just hits these two major roadblocks.
Okay, what's the first one?
The first wall is heterogeneity. Just think about that skull and brain example. You've got really rigid, dense bone fibers literally right next to extremely soft, delicate neural tissue.
Yeah, totally different materials. Their optical properties, their mechanical properties, miles apart.
Exactly. And that leads straight into the second wall, the classic trade-off between resolution and scale.
Right. You need that super fine submicrometer resolution to see the details.
But to get that across a large volume, you typically need to section the sample, cut it into thin slices iteratively.
And that's where the problems start with heterogeneous tissue.
Absolutely. If you try using traditional methods like a mechanical microtome to cut both bone and soft tissue at the same time, forget it.
What happens?
The bone resists the blade, it's tough. But the soft tissue gets distorted, compressed, maybe shrinks unevenly. It's basically guaranteed to deform.
So you lose the true structure, especially at the boundaries.
Precisely. Those critical interfaces, like where a tiny blood vessel crosses from the skull through the membranes, the meninges, and dives into the brain, that delicate connection gets destroyed by mechanical cutting.
Okay, so mechanical cutting is out for this kind of in situ work. What about chemical methods, tissue clearing?
That has its own issues. Clearing agents often struggle to make materials as different as bone and brain transparent uniformly. You might clear the brain okay, but the bone remains opaque, or vice versa. It's very difficult.
So this brings us to the focus of our deep dive today. Our sources describe a really significant breakthrough, a smart robotic system.
Yeah, one that basically throws out the mechanical scalpel altogether.
And uses light instead to do high-resolution histology in situ, right there in its original place.
Exactly. And the core of this breakthrough is a laser technique called spatiotemporal focusing, or STF.
STF, okay.
It's described as a kind of shortcut, maybe the ultimate shortcut, because it manages to preserve that crucial anatomical structure, especially at those difficult interfaces between different tissue types.
And it delivers this detailed volumetric data on a scale that just wasn't possible before.
Right. It's a really clever fusion of technologies.
Okay, let's unpack the system itself first. What are we actually looking at? The papers describe it as having this dual identity. It's both a microscope and a precision cutting tool.
That's really the heart of it. It integrates two main components very tightly. First, you have a diffraction-limited scanning multiphoton microscope. That's your imaging system.
Multiphoton. Okay, good for deep tissue imaging.
Exactly. And to handle that heterogeneity we talked about, it's smart. It actually uses different imaging modes depending on the type of material it's looking at.
Ah, so it changes how it sees based on whether it's looking at bone or brain.
Precisely. Specialized eyes for specialized materials, as you put it. For instance, for the rigid stuff, like the collagen-rich bone, it uses backscattered second harmonic generation, SHG. It's great for ordered structures.
Okay. And for the soft tissues, the neurons, the blood vessels.
For those, it relies on fluorescence, either intrinsic fluorescence from the tissue itself or, more commonly, fluorescence from specific labels that have been introduced, like dyes marking the vasculature.
Got it. So it images, figures out what's bone, what's soft tissue, and then it moves to the cutting step.
Right. And the cutting. Well, that's where the real engineering marvel comes in.
Right.
It uses temporal focusing-based laser micromachining.
Temporal focusing. And this uses light as a scalpel. How does that even work? I mean, wasn't that much energy just cook the surrounding tissue?
That's the clever part. It uses extremely short, like femtosecond short, laser pulses. These are incredibly intense bursts of light.
Femtosecond, that's a millionth of a billionth of a second, right? Extremely short.
Vanishingly short. And when these intense pulses hit the exact focal spot in the tissue, the energy density gets so high that the tissue absorbs the light through nonlinear absorption processes.
Nonlinear absorption, so it's not just simple heating like leaving something in the sun.
Not at all. This nonlinear absorption generates a highly localized plasma right at the focal point. Think of it like creating a tiny, contained spark or gas bubble.
Plasma. Okay, so it's not a thermal burn spreading outwards. It's more like a tiny controlled explosion
Right.
confined precisely to the focus.
That's a great way to think about it. The official term is plasma-mediated ablation. Ablation just means material removal.
And because that plasma is confined to the tiny, diffraction-limited focal volume of the microscope.
The material removal, the cut, is done with the absolute highest possible precision. We're talking diffraction-limited precision. The cut is literally as fine as the microscope can resolve.
Amazing. But you mentioned earlier there was a problem with previous attempts at this kind of thing.
Yeah, the big hurdle was speed. Plasma-mediated ablation with a tiny point focus works, but it's slow. Painfully slow if you want to map something as large as an entire mouse brain.
Right, you'd be there for weeks, maybe months.
Exactly. So the challenge was, how do you increase the speed, the efficiency of removal, without sacrificing that critical axial precision you need for serial volumetric imaging? You still need clean cuts, layer after layer.
And that's where spatiotemporal focusing STF comes in.
That's the core innovation right there. STF solved the speed problem by fundamentally changing the geometry of the laser focus used for cutting.
Okay, how does it do that? How does STF take those femtosecond pulses and make them cut faster?
It's a really elegant piece of optical engineering. Instead of focusing the laser pulse down to that traditional tiny round spot, a Gaussian focus, which is like drilling with a microscopic pin,
Yeah.
the STF system first takes the ultra-short pulse and stretches it out in time using a diffraction grating. This makes the pulse duration maybe 10 times longer or even more.
Stretches the pulse.
Okay.
Then what?
Then it uses a special optical setup, what's called a 4F system, involving a cylindrical lens and the high numerical aperture objective lens to reconstruct that stretched pulse at the target.
And what shape does the focus beam take now? It's not a point anymore.
No. It reconstructs as a line-shaped femtosecond pulse right there on the imaging focal plane.
A line instead of a dot.
Exactly. Think of it like switching from that tiny pinpoint drill bit to a very fine, very sharp laser chisel.
Ah, I see. So you're removing material along a line instead of just at a point with each pulse.
Precisely. By distributing the laser energy efficiently along this specific line, the paper mentions dimensions like 71 micrometers long, but only about 0.6 micrometers wide, rather than concentrating it all in one spot.
You get more bang for your buck, pulse-wise.
Massively more. The sources state it results in a 100-fold increase in single pulse ablation efficiency. 100 times.
Wow. Just by changing the shape of the cut from a point to a line.
It allows the system to slice through an entire plane of tissue much, much faster than before, while still using that precise plasma-mediated ablation.
And the precision is still maintained. What about the depth of the cut?
Still incredibly precise. The measured ablation spot dimensions they report are, let me check. Yeah, delta around 0.62, 0.0 delta, the line length is 71 delta. And crucially, the axial depth delta, Zieving deltas, is only about $6.1.
6 microns deep. That's thinner than most human cells. So you're really getting clean, thin slices removed optically.
Exactly. This line ablation seems to offer the best possible trade-off between boosting the removal efficiency, the speed, and maintaining that critical spatial precision, especially in depth.
Okay, faster, precise cutting, sounds great. But there's always a but in complex systems like this, right?
Which ways, and this one's a doozy, the bubble trouble.
Bubble trouble.
Remember, to get that super high resolution, the microscope's objective lens needs to be immersed in a fluid, usually something water-based. It needs that immersion medium.
Right, high numerical aperture objectives need immersion.
But what happens when you fire these incredibly intense femtosecond laser pulses into a water-based medium during ablation?
Uh-oh. You're basically boiling the water instantly at the focus.
You got it. You generate cavitation bubbles, tiny pockets of steam and plasma.
And I bet those bubbles don't just disappear in Iceland.
Nope. They tend to accumulate, they stick to the objective lens, they stick to the sample surface, and once they're there, they scatter the incoming laser light like crazy.
Blocking the beam, both for imaging and for further ablation.
Exactly. The sources say this bubble accumulation can stop the continuous machining process dead in its tracks within seconds.
Seconds. So you could never run this for the days needed to image a whole brain if bubbles kept stopping it.
Absolutely not. So they had to engineer a solution, and it was quite clever. A miniature, high-speed, closed-loop flow system.
Like a little plumbing system built right into the microscope stage.
Pretty much. A sophisticated microfluidic system designed specifically to constantly flush the area right under the objective lens. It washes away those laser-induced cavitation bubbles and any little bits of sample debris immediately as they form.
Keeping the optical path clear for continuous operation. Very neat.
It was essential for enabling long autonomous runs.
Okay, so the bubbles are handled by the flow system. But that brings up another potential stability nightmare, doesn't it? The fluid itself, the immersion medium.
Mm-hmm.
If you're running this for days, maybe 96 hours as mentioned, surely things like evaporation start to matter. Doesn't the composition of the fluid change?
You've hit on one of the most critical and honestly one of the most brilliant parts of the robotic control system they developed.
Yes, the immersion medium is crucial, and maintaining its properties is paramount.
Why is it so critical?
Because the refractive index, or RI, of the immersion medium directly affects the focal length of the objective lens. If the RI changes, the laser focus shifts up or down.
And over days, especially with a flow system, the more volatile components, like water, are going to evaporate faster than others, right? So the RI will drift.
Precisely. They chose their medium carefully, a mix of 60% DMSO and 40% water, with a buffer because it had a good balance of low viscosity to help flush bubbles and a relatively high ablation threshold. But they knew RI drift would still happen.
How bad could that drift be?
Bad enough to completely ruin the experiment. The paper notes that even a small change in RI can shift the laser focal spot vertically by over 100 micrometers.
Whoa. When you're aiming for cuts only 6 microns deep, a 100-micron focal shift is game over. Totally unacceptable for submicrometer precision histology. So they couldn't just hope for the best. They needed active control.
How did they achieve that? Did someone have to manually check the RI every hour?
That's the ingenious part. They built an artificial vision-based feedback control system. Completely automated?
Artificial vision. How?
Okay, get this. They route the circulating immersion fluid through a standard digital refractometer, which measures the RI continuously.
Standard lab gear. Okay.
But instead of having a person read the display, they aimed a simple USB camera right at the refractometer's digital readout screen.
Seriously, a camera watching the screen.
Yep, cheap and effective. Custom software then analyzes the image feed from that camera in real time. It uses image processing to read the RI value off the screen.
That's surprisingly low-tech, but clever.
It works. The software compares the measured RI to the target value. If it detects drift, usually indicating water has evaporated, increasing the DMSO concentration and thus the RI, it automatically triggers a microperistaltic pump.
To do what? Add more DMSO?
No, to add tiny, precisely measured amounts of water back into the closed-loop system to bring the RI back down to the set point.
Wow. A self-correcting system based on a camera watching a screen. And it was stable enough.
Incredibly stable. The data shows that over a 96-hour continuous run, this feedback loop maintained the refractive index with a coefficient of variation of just 0.000016.
That's astonishingly stable. Basically negligible drift over 4 days.
And that level of automated stability control is absolutely fundamental to making multi-day, autonomous, high-precision histology possible.
Okay, let's recap quickly. We have this system that uses multiphoton microscopy to see different tissues. It uses spatiotemporal focusing to create a line-shaped laser pulse for fast, efficient cutting via plasma ablation. It beats the bubble trouble with the flow system. And it maintains perfect focus over days using a camera watching a refractometer to control the fluid's refractive index. That's the hardware and the basic physics sorted.
Pretty much, yeah. A very sophisticated setup.
Now we get to the smart part. You mentioned the robotic control pipeline. This is what manages the sheer complexity of cutting through different materials, layer by layer, autonomously for days.
Right. Autonomy is the name of the game here. The software, largely based on open source tools, actually, is what orchestrates everything. The entire volume to be imaged and ablated is conceptually divided into many thin, partially overlapping layers.
And it works through these layers iteratively.
Exactly. It follows a core 4-step workflow loop for each layer. Explore, scan, analyze, ablate. Explore, scan, analyze, ablate. Over and over.
Okay, what happens in each step? Explore sounds like a quick look.
Yeah, Explore is a rapid, low-resolution surface imaging pass. Just to get the lay of the land for that layer and define the specific region of interest, the ROI, that needs high-res imaging and ablation.
Then scan is the detailed picture.
Right. Scan is the high-resolution, multiphoton imaging of the volumes of interest within that ROI, using those material-specific modes like SHG for bone and fluorescence for soft tissue.
Got it. The analyze. This sounds like where the intelligence comes in.
Absolutely. This is a crucial online processing step. The high-res scan data is immediately processed things like downsampling for speed, stitching together adjacent image tiles, and critically, segmentation and voxel classification.
Voxel classification. So it figures out for every tiny 3D pixel or voxel, whether it's bone or brain or something else.
Precisely. It needs to know exactly what material it's about to cut because, as we discussed, soft brain tissue and hard skull bone need very different ablation parameters to be removed cleanly and efficiently.
Right. Different laser power, different exposure times.
Exactly. The paper gives examples like gelatin, used as a soft tissue model, needed about 10 joules per square centimeter of laser fluence, whereas the rodent skull needed much less, only about 2 JCMS. The software uses the classification map to assign these material-specific machining parameters, voxel by voxel, or region by region.
So it tailors the cut dynamically based on it sees it's cutting.
Millisecond by millisecond, potentially. It adapts the laser on the fly.
That's seriously smart. Okay, so analyze figures out what to cut and how to cut it. Then comes ablate.
Which is the physical removal using the STF laser line focus. But even here, there's more intelligence. The system doesn't just raster scan the laser back and forth blindly.
It has to plan the cutting path efficiently, right? Especially across a potentially large and complex shaped area for that layer.
Exactly. It treats the required ablation path for that layer as a classic computer science puzzle. The traveling salesman problem, or TSP.
The TSP. Finding the shortest route to visit a set of cities. How does that apply here?
The software considers all the distinct regions, the voxels or groups of voxels, that need to be ablated on that layer as the cities the laser focus needs to visit.
Oh.
It then uses a well-known optimization algorithm, specifically a 2-opt algorithm mentioned in the sources, to calculate the shortest possible path for the laser scanner to travel to cover all those target regions.
So it minimizes wasted movement and time, maximizing the cutting speed for that layer.
Precisely. Efficiency is built right into the physical path planning of the laser beam.
Clever. But what about errors? What if the ablation isn't perfect? If it misses a tiny spot, leaves a bit of tissue behind from the layer it just tried to remove?
That's a critical point, because any residual tissue, even microscopic bits left on the surface, will severely scatter light. It would ruin the quality of the scan for the next layer down and likely mess up the ablation of that next layer too.
So perfect removal of the current layer is essential before moving deeper. How does it ensure that? Quality control.
Yes, and that's where the explore step comes back into play, but in a different way. After an ablation step, the system often performs another quick explore scan of the newly cut surface.
To check its work.
Exactly. It uses that low-res image for ablation quality control. The analysis software looks for any signs of residual tissue that should have been removed.
And if it finds some, does it just flag an error?
No, it tries to fix it automatically. If residual tissue is detected, the system halts the normal workflow progression and re-enters the ablate mode specifically for refinement to target those missed spots.
Does it just hit them again with the same laser settings?
Not quite. The sources mention something interesting here. Residual tissue that's slightly below the ideal target plane, maybe because the first pass wasn't perfectly even, is harder to remove.
Right, the laser might be slightly out of focus for it.
So for these refinement ablations, the system actually calculates and applies an adjusted higher laser fluence, and this adjustment is depth dependent. It uses a formula, FF0E Se, which essentially means the further the residual target Z is below the intended ablation plane, the higher the exponential factor applied to the base fluence, FS-Sieber. It ramps up the power to ensure it blasts away those deeper remnants.
Wow, that's relentless self-correction. It really ensures a perfectly flat, clean surface for the next layer.
It has to, for the whole process to work over millions of cubic microns. And there's one more layer of monitoring, too.
What's that?
Continuous hardware health monitoring. Throughout these multi-day runs, the control software is periodically pinging critical hardware components, checking the imaging laser power stability, checking the timing and triggering signals between the lasers, scanners, and detectors.
Making sure everything is still working correctly and in sync?
Yeah. If it detects any instability, like a drop in laser power or a timing mismatch, it can trigger an alert or even automatically pause and attempt to reacquire the image for that section, trying to ensure flawless data acquisition over the entire marathon run.
Okay, this whole system is incredibly complex, integrating optics, fluidics, robotics, and intelligent software control. Let's talk about the payoff. Did it actually work? What results did they get?
The results reported are pretty spectacular, really validating the whole approach. They demonstrated it on arguably the perfect test case, imaging the entire craniocerebral vascular network of a mouse right through the skull.
From the outside of the skull all the way down into the brain tissue.
Exactly, down to the white matter. And they achieved impressive ablation rates doing it. Let's see. Soft tissue, modeled by gelatin, was ablated at about $7.7 times 105 milliterature per second.
Okay, nearly 1,000,000 cubic microns a second. And the hard tissue, the bone.
That was slower, as expected, around 1.4 times 105 milliterature per second, but still very usable. And critically, they verified that the collateral damage, the unintended damage below the cut surface, was confined to within just 5 micrometers below the last ablation plane.
Very localized removal, minimal damage to the underlying layer you want to image next.
So that's the goal.
And the final reconstructed volume, how big was it?
It was massive for this level of resolution. They successfully imaged and computationally stitched together a volume of $5 4 times 4.2 times 1.8 text millimeters 3 class. That's a huge chunk of the mouse head, rendered as a single coherent 3D dataset.
And the resolution was maintained throughout that volume.
Yes, uniform submicrometer resolution. They quote voxel sizes of $0.35 times 0.35 times 1.0 mu 33, so incredibly detailed.
What specific biological features could they see with this method that would be difficult or impossible otherwise? You mentioned interface preservation earlier?
Right, that was the key demonstration. The images clearly showed intricate details like the fine fibrous structures within the bone itself and even tiny microvessels running within cavities inside the skull dome.
Wow, vessels inside the bone.
Yeah. But the real showstopper was visualizing the boundary between the skull and the brain. They could clearly distinguish a blood vessel running within the meninges, the membrane covering the brain, from a vessel running just underneath it, on the surface of the brain itself, a pile vessel.
And they could see the separation.
Perfectly. They could resolve the thin layer of meningeal collagen separating those two vessels, which were physically very close but functionally distinct.
And that delicate separation, that interface, is exactly what gets mangled by traditional mechanical sectioning, right? Because the bone and soft tissue deform differently.
Precisely. Trying to slice that interface mechanically almost always leads to tearing, compression, or artificial gaps because the hard bone and soft meningeus brain react so differently to the blade. This all-optical method completely avoids that physical distortion.
So comparing this to other ways of getting 3D anatomical data.
This all-optical serial 2-photon microscopy, powered by STF and the smart robotic control, really stands out. It seems unique in its ability to achieve that submicrometer resolution in situ imaging across the intact craniocerebral system, handling both bone and brain.
What are the core advantages then?
Minimal sample preparation is a big one. No harsh clearing chemicals, no embedding in wax, minimal sample deformation because the ablation is non-contact, it's just light. And the crucial one, preservation of the anatomical integrity, especially at those tricky interfaces between different tissue types.
That seems huge for understanding complex structures accurately.
It is. The main trade-off, however, is important to state.
That's the downside.
The method is destructive in the sense that the ablated tissue is vaporized by the laser. It's gone. You don't retain the physical tissue slices for other kinds of analysis, like histology stains or genetic tests, which you might do after mechanical sectioning.
Ah, right. You get the 3D structure image, but not the physical substance itself for follow-up.
Exactly. It's a choice between pristine structural data in situ or retaining the physical sample potentially with some distortion.
Okay, so let's try to synthesize this. We've journeyed through a complex system that leverages some pretty specialized physics spatiotemporal focusing to turn a femtosecond laser into an efficient, line-shaped cutting tool.
Combined with really sophisticated adaptive autonomous software control, the robot managing the fluidics, the imaging, the material-specific ablation, the path planning, the quality control.
Right, and the result is the ability to optically dissect and digitally reconstruct these large, complex, mixed material biological volumes like the skull and brain together that were essentially inaccessible at this level of detail before.
It overcomes the limitations where mechanical cutting or simple chemical clearing just couldn't maintain the structural truth, especially at those critical boundaries.
So for you listening, what's the key takeaway here? It seemed like the frontier in getting this kind of incredibly detailed scientific data isn't just about building better optics or more powerful lasers anymore.
That's right. It's increasingly about the smart integration. fusing precision manufacturing techniques like femtosecond laser ablation with highly adaptive, self-correcting, intelligent machine control systems. The technology has to almost think for itself to manage the sheer complexity and scale of biological systems.
The future is integrated and autonomous. Now, building on that trade-off, you mentioned the fact that this brilliant all-optical method destroys the tissue it removes. Here's a final provocative thought for you to consider. Given how important it often is to keep the tissue sample for other types of analysis, biochemical, genetic, proteomic, what kind of future technology might be needed, could we imagine a parallel or maybe a hybrid approach? Something that combines the incredible structural integrity you get from this all-optical imaging, but somehow integrates it with the sample retention capabilities you might get from, say, advanced mechanical sectioning or perhaps some non-destructive chemical analysis done layer by layer. What will it take to achieve the ultimate goal? Multimodal, high-resolution anatomical mapping of perhaps an entire body, where you get both structure and substance.
That's a fantastic question. That pursuit of true, multimodal, non-destructive, high-resolution reconstruction at scale, that really is the next great challenge, isn't it? Combining the best of all worlds.
Something for us all to think about. Until next time, keep digging deeper.